This is a GCSE Biology presentation looking at transport in plants. To get started, what you've been presented with is a diagram that represents a cross-section of a leaf. So if you took a leaf and sliced and looked along the edge of it, what would you see inside? What you have to do is identify all the different labels from 1 to 10. At this point, pause the presentation, try your best to come up with as many answers as you can, and whenever you're ready you can restart the presentation and go through your answers. So pause now. Well, in terms of answers, so what we've got in the top layer is called an epidermis. You can refer to it as the upper epidermis. Remember, part of the upper epidermis is that cuticle layer, which is the orange layer along the top, and that is a waterproof layer that prevents water loss. Number 2 is the palisade mesophyll. Remember, that's got all the chloroplast to maximize the rate of photosynthesis. Number 3 is the spongy mesophyll layer with the air spaces to increase the rate of gas diffusion. We've got the lower epidermis. We've got a guard cell with the stomata, which is the hole in the center, a guard cell on the other side of the stomata, so there's one guard cell on either side, and we've of course got the xylem, the phloem, and they make up the vascular bundle. Now, if that doesn't make sense, please go through the presentation on plant structures and it'll take you through it in more detail, and then of course you can come back to this presentation when you're ready. In this lesson, we're going to look in more detail about the structure of the xylem and the phloem vessels, and we're going to talk about processes called transpiration and translocation. A xylem vessel and phloem vessel are the two main vessels found inside a plant, in particular in a plant stem, but they're also found in plant roots. The xylem is a hollow tube. It has lignin around its outside to help strengthen those cells, and it's involved in the movement of water. Notice there are no layers in between the cells. There are actually gaps so water can freely flow from one cell to the next. Remember, the xylem also carries up mineral ions. The phloem, which is on the far right hand side, is a slightly different structure because unlike the xylem, it's made up of living cells. Because it's made up of living cells, it also has beside each of the phloem tubes or sieve tubes, you have what are called companion cells. And companion cells are necessary to supply all of the phloem with its energy for transporting substances. Without those companion cells, the cells in the phloem would die. In between the phloem, we have what are called sieve plates. You can also notice at the bottom the structure in the root and in the stem, that we have a very different format. In the root, we have a kind of cross in the middle, which is the xylem, and then in 4 bunches around the outside we have the phloem, whereas in the actual stem we have the tubes basically side by side in combination of pairs, with the phloem around the outside and the xylem on the inside of the stem. So in terms of transport through a plant, there are 2 main transport pathways. We talk about translocation and we talk about transpiration. Translocation is linked to the phloem. Transpiration is linked to the xylem. In translocation, we talk about the movement of dissolved sugar. In transpiration, we talk about the movement of water, but that does carry with it mineral ions. But transpiration is focused on the water part. Just remember, the mineral ions will move up the xylem with it. And in translocation, we have movement up and down through the phloem. Whereas in transpiration, we only have movement up the xylem through the stem. Let's look at the process in a little more detail. In translocation, we've got photosynthesis occurring in leaves. And we know it occurs in leaves, mostly in the palisade layer, because that's where we have the highest number of chloroplasts, which absorb the most light intensity, which means it has the fastest rate of photosynthesis. In photosynthesis, remember, we take carbon dioxide and water, and in the presence of sunlight energy, that is converted into glucose and oxygen. At the moment, we're not worried about the oxygen, we're just going to focus about the glucose because that's what's linked to translocation. When we have glucose, all cells in the entire plant need that glucose for respiration. They've all got their own mitochondria. They need to have the glucose as well. In particular, think about those cells that are underground in the roots, which don't have access to sunlight energy ever. Here's an important thing. When we talk about movement through translocation, we talk about dissolved sugar. You shouldn't talk about dissolved glucose, because it's not actually glucose that moves. The plant turns the glucose into a different sugar, and that sugar moves through. And if you do A-level biology, you'll find out about that in more detail, and there are presentations for A-level biology on photosynthesis should you wish to look them up. But for GCSE, we can simply say that photosynthesis results in the production of glucose, and glucose is needed by all cells, and as a consequence, dissolved sugars are transported through the phloem up and down the plant, and that's known as translocation. Transpiration is the more common process to find on a GCSE paper, but they still can test you on translocation, so make sure you know it well. In transpiration, we talk about the xylem vessel. We're talking about the movement of water. But remember, in that water we have dissolved mineral ions, so the mineral ions will move as well. But we focus on the water for transpiration. Carbon dioxide gets into the plant through the stomata. As a consequence, when the stomata are open, remember if they're open, it means the guard cells are turgid. If you're not sure about the term turgid, go and look at the plant structure presentation before you go any further. But when we have turgid guard cells, the stomata are fully open. With the stomata fully open, we have the maximum rate of carbon dioxide moving in by diffusion, but we also have the maximum rate of water evaporating out the stomata. As a consequence, that water has to be replaced, and it's replaced with water from the xylem vessels. Because there are hydrogen bonds in water molecules, they are attracted to each other, and we have what's called cohesion. And cohesion can be seen very easily. If you put water in a very narrow tube, you get this meniscus effect where you have the water, instead of being perfectly level, creates a little U shape. And that's where we've got cohesion going on. But cohesion results in all the water molecules being attracted together. So as water is sucked out of the xylem, the water is then pulled up through the xylem from the water at the bottom near the roots, and then water can move by osmosis through the root hair cell, through the rest of the root, and all the way across to the xylem at the bottom of the plant. So at the bottom near the root we've got osmosis. Then in the xylem vessel we talk about the cohesion, because of the hydrogen bonds between the water molecules pulls those water molecules up. The reason they're pulled up is because the water has evaporated out the stomata and has been replaced by water at the top of the xylem. And that process is known as the transpiration stream or transpiration. Remember, it's osmosis at the bottom, it's cohesion in the xylem, and it's evaporation of water at the stomata. Now we can actually change some environmental conditions, and that will affect the rate of transpiration. There are other things that we can do that will also affect the rate of transpiration. For example, the number of stomata on the underside of a leaf. The more stomata, the more holes, the more water can evaporate out, and therefore we have a higher rate of transpiration. But you need to specifically know about how the rate of transpiration is affected by a number of environmental factors. Temperature, humidity, the wind or air movement, and light intensity. What I'd like to do is take 2 minutes, pause the presentation and answer the question, how would you change each of those variables, so increase or decrease them, so that you would increase the rate of transpiration? Pause now, come up with an answer, maximum 2 minutes, and then you can restart the presentation. Well, in order to increase the rate of transpiration, we want water to evaporate as quickly as possible out the stomata. So we do that by increasing the temperature. We do that by decreasing the humidity. Remember, the humidity is the amount of moisture in the atmosphere. If there's lots and lots of moisture in the atmosphere, then we slow the rate at which water will evaporate out. If the atmosphere around the plant is very, very dry, then we'll have very, very fast rate of transpiration as the water evaporates out very quickly. In terms of air movement and wind, we have an increase in the air movement will increase the rate of transpiration, and light intensity, also increase in light intensity will increase the rate of transpiration. The easiest way to remember it is try and think about if your clothes were outside and you put them on a washing line to dry, what conditions would make your clothes dry the fastest? Because when we're drying our clothes, we want the water to evaporate from our clothes in the same way that during transpiration, the water should evaporate out the stomata. So we look for the environmental conditions that will increase the rate at which water will evaporate, and those environmental conditions will increase the rate of transpiration. Obviously, the reverse is true if we want to decrease the rate of transpiration. It's not impossible in the exam that they'll give you a question about some kind of investigation into transpiration. So you need to make sure you're aware of what it means by independent variable, and that's a factor that's deliberately changed in an experiment. For example, the temperature, humidity levels, the air movement, or the light intensity. You can also find investigations into transpiration where they've changed the number of stomata, the size of the leaf. There's a lot of different variables that can have an impact. But remember, the independent variable is the one that is deliberately being changed and the values are decided before the experiment starts. The dependent variable is the variable that we measure and record during the experiment. It's essentially the results. And quite often for transpiration investigations, it'll be some kind of measure of how much water has been lost from the plant, either by measuring the before and after mass or in some way measuring the volume of water that's been absorbed by the plant. And it's a specialist piece of equipment that we can use to measure that volume of water. A petometer is a device that lets us measure the rate of transpiration. If you look at the picture on the right hand side, that's the more traditional petometer, and what you have is a plant which is got a cork, so the only way that water can get out is by going through the plant. If you follow all the way down along the tube, you'll see there's a number of little dashes like a ruler measurements and a small white bubble. That small white bubble will move as the plant draws water from the tubes. There's a second tube with a little tap at the bottom in the middle of the picture, and that little tap at the bottom simply is when we're ready to reset it and we want the bubble to go back to zero, we can simply open that little valve and the water goes in and it pushes the bubble back to zero. And as a consequence, we can set this up, close the tap, leave it for a certain amount of time, and we can measure how much water the plant has drawn up through its stem, and we can use that as a measure of the rate of transpiration. So you need to be able to actually calculate the rate of transpiration, and to calculate the rate of transpiration, what you would normally do with the petometer is you would take the distance that the bubble moved and you divide it by the amount of time it took for the bubble to move that distance. Quite often you'd have to take this information from a graph, and remember that means you're looking for the gradient, which is how steep the graph is. We can calculate that by working out the difference on 2 points on the y-axis and then the difference on 2 points in the x-axis. Be careful when we're talking about rate of transpiration in any experiment, don't use the term amount. If we're talking about solids, we talk about the mass of material. If we're talking about liquids or gases, we're talking about volume. And it's different for every experiment, but the rate is always a measure of how much something is occurring in a certain amount of time. So for example, the number of grams used per second or the number of grams produced per second, the number of milliliters used per minute. the number of people going past per hour. It's always the amount of things that happen in a certain amount of time. And you might have to do it from a graph, or you might have to do it from a calculation. And if it's a petometer, it's always the distance moved divided by the time taken. The last thing you have to do to finish off is you've got to do a don't stop activity. What you have is 2 minutes worth of time, and you are not allowed to repeat yourself, pause, or make a mistake in anything you're talking about. And you're going to talk about the material from this presentation for 2 minutes. You can talk to a family member, a pet, mirror, or even yourself. You can use your internal voice, your external voice, whatever you're happiest with. Once you've done your 2 minutes, go back through the presentation, see if there's any important information or any keywords you could add, and try and repeat the process again, keeping to the 2 minute time, but adding that extra content in.